All right, welcome to Science 30, Unit A, Chapter 2, Genetics, and this is the last lesson in the chapter on genetic technologies. So by the end of this lesson, you should be able to describe in general terms genetic engineering and its application to gene therapy and the development of genetically modified organisms. We have the textbook pages down there that we'll be referring to. So first technology, genetic technology we're looking at is DNA fingerprinting. This was developed back in 1985, and it allows you to take DNA samples from different people and compare them to look for similarities and differences. So this has been used to solve crimes and also in paternity testing, figuring out who the parents are. Any two people in the world will have about 99.9 the same DNA, but that's that difference between people is what fingerprinting looks at. So it takes the pieces of DNA that are different between people and compares those different parts. There's a great video here that you can watch that goes into more on DNA fingerprinting. So how does DNA fingerprinting work? Well, first off, we use the PCR or polymerase chain reaction, which is a reaction that takes DNA samples and amplifies it, makes a whole bunch of copies of it. So you get a whole bunch more fragments of the DNA, and their fragments are separated through this negatively charged gel. So you don't have to worry about how that happens, but it's basically the shorter segments tend to move a little faster through the gel, and they separate into this very characteristic pattern here, these fingerprints. And then we can use the patterns to compare. So in this question here, you have 4 sets of parents and 1 child, and you're trying to figure out who that child belongs to. Okay, remember, children get all their DNA from the 2 parents. So the child's only place the child can get its DNA is from the parents, so they have to match 100%. So if you look, you can see this child has matches the parents B. All their DNA has been passed on to that child. So that child only has DNA from that parent. You can see that how it quite nicely matches. Okay, so we mentioned PCR, polymerase chain reaction. It's actually used quite a lot of different ways. So DNA fingerprinting is one of them. Right now, it's actually being used as part of the COVID-19 test. So when someone's infected with, or to see if somebody's been infected with the coronavirus, this PCR is actually part of the testing process now. So what it generally does, the steps are here, what generally happens, you get the swab in the back of the nose. They get any mucus or whatever on there, but what they're also looking for is bacteria or viruses that might be sitting back there too. So they then put that sample through a bunch of processes to get rid of everything except the RNA, which is a substance very similar to DNA. It looks very similar. It's made of almost the same bases except for one, but it functions a lot of the same ways. And then they use that RNA and the DNA to kind of stick onto it, to the RNA that was from the virus. Okay, so they're looking at that virus contains RNA, and they're looking at it, and they're going to make a copy of that RNA using DNA, and then they're going to amplify and make a whole bunch, millions and millions of copy of that DNA that matches the virus RNA. And that'll help them figure out how much of that virus RNA was there and if it's a positive test or not. There's a couple of videos on here if you're interested in how that test works more specifically. You don't really need to know the specifics around this test at all. It's just quite interesting that the PCR test is involved in this. And just another little kind of tidbit here, that RNA sequence. So again, like RNA is basically the same thing as DNA. You can think of it that way. It's just another way that our genetic information is passed on, can be passed on through RNA, and viruses have RNA in them as well. Anyways, this RNA from the virus that causes COVID-19, it is about 30,000 bases long. Remember, bases are like the A, T, C, G. It's just slightly different in RNA. And it contains the instructions for making 29 different proteins. And there's a little diagram of that. And this is figured out really early on in the outbreak. And now we can figure out these sequences quite quickly with the technology that exists these days. Or it might have taken years before, could be done very fast. Anyways, moving on. So the next topic is transgenics, or genetically modified organisms. So genetic engineering refers to the modification of genetic material through the actions of humans. So we manipulate the DNA, we might change the DNA, we might build new DNA by modifying old DNA. You might splice two pieces of DNA together. They might be DNA from or three or four different organisms, and you put them together. That's recombinant DNA. So there's a lot of types of different genetic engineering that occurs. So when you're working with recombinant DNA or looking at recombinant DNA, there's four main things you need to have. So you need to have that original DNA, or in this case, a plasmid, a circular DNA from that bacteria. The gene that you want to put into that DNA. You need restriction enzymes to cut that plasmid. And then you need ligase, which is like glue enzymes, to kind of snap it back, this new piece, into that plasmid. Okay, so that's if you're cutting and recombining DNA. Those are some of the pieces you need. So there's an example here of a genetically modified organism, or GMO. Example is golden rice, and it has DNA from 3 other plants and 1 fungus. It's pretty cool. Then this allows this rice to have an increased amount of iron in it and vitamin A content. So you can see here from beans, there's the gene that came from beans. So the rice DNA was spliced, cut, and this piece of genetic material from beans was ligazed or glued right in there. Right? And this helped to increase the iron content of that rice. Why would you want to increase the iron content of rice? Well, if rice is being fed to people in poverty-stricken areas, this is a great way to boost the amount of protein and how far that rice can go for human health. It might just save that many more people if there's more protein content in that food. So there's a lot of controversy around GMOs, and it's constantly in the news, and it's good to be really interested and inquisitive in this idea because it seems so foreign. This infographic is talking about, are GMOs safe, and they're saying yes in this infographic, that there were this is referring to a report that says they were, and they looked at over 900 studies and publications that examined them. So by safe, though, what are they looking at? I believe in this case they were looking at risk to human health. Okay, so it's really important to look at what is the question and who does the question affect, and ask as many questions as you can about the question that they're studying. So are GMOs safe? Okay, so they're saying all these studies, over 900 studies, say that they're safe for human consumption. But there's a lot of other pieces to the GMO puzzle. So some of the questions are, especially when we're talking about genetically modified food and plants, who owns them? Right? When you've created a seed for a type of canola and this company owns it, if it falls onto your property, can you regrow it, or are you going to be charged? Right? Who owns that plant? Should they be patentable? Can you patent life forms? Right? There's this idea of possibly resurrecting extinct species. So you take a DNA from some extinct species and put it into a species that is existing now, and recombining their DNA to see what kind of things you get. They're talking about bioweapons, but also, like, you got to look at the safety of the environment too, the ecosystem. So it might be okay for humans' health for these foods, but how is it affecting other pieces of our environment? If you're creating a pesticide-resistant wheat, how is that affecting the ecosystem? So it's keeping the pests away, but is it affecting other organisms in that environment? Yeah, there's just a lot of questions around GMOs and a lot of controversy still. Gene therapy. Okay, so this is another use of genetic technologies. This is where you are actually altering genes in human cells to cure or treat a genetic disorder like cystic fibrosis, diabetes, Parkinson's. So basically, you want to get into the human cells, into the DNA, take out the bad part, put in a better part, is a way of thinking about it. Yeah. So there's a lot of trials going on right now in humans. And when they do trials, they focus on the somatic cells. These are the body cells and correcting to the disease in the body cells. This does never prevent the offspring from still getting this disease because they're not doing this therapy with the gamete cells, right? They're not going into egg cells or sperm cells, which is what is going to be passed on to the next generation. That's actually banned in Canada and most other countries as well. Right now, we're not allowed to alter that DNA in egg and sperm cells, but we can do trials in human cells to try and fix or change the DNA in human cells. You can probably think of a lot of different ethical issues around gene therapy. And if you start doing a little research online, you probably come across pretty cool stuff. There's a good video here on gene therapy if you want to check that out. And there's just some pros and cons here on a couple slides. I'm sure you can think of some more, but here are some to get you thinking about of recombinant DNA. Some of the pros: production of human products without side effects. So something like insulin, life-saving drug. We can put that gene for insulin creation into bacteria and get the bacteria to make insulin for us, right? Then we can use that to save lives for people. Production of disease-resistant strains of plants for agriculture. So you put this gene for pesticide resistance or herbicide resistance into the plant that you want to grow, and then you can put herbicide everywhere else, get rid of all your weeds, but the plant you want to grow keeps growing, right? Combine characteristics of two organisms into one. So you got two things. You want to see a green bunny, right? No, I don't. Yeah. Very specialized form of selective breeding. So this really speeds up our selective breeding process too. What could take many, many generations, hundreds of generations to get to, we could do a lot quicker through recombinant DNA. And then gene therapy is a definite possible pro. Some cons of recombinant DNA. Some people believe that we're playing God because we're selecting those DNA and genes and putting them in to organisms to change the way they're going and modifying them. Example, or sometimes people refer to this as eugenics. We may use it to select genetic information in offspring. There's a worry about that. It is a very expensive procedure, so it's at that elite level, not everybody can partake. We're not sure about the long-term effects on other organisms and the ecosystem. And it can lead to development of resistant viruses and bacteria and plasmid transfers. Just one other piece in Science 30, you're going to look at perspectives frequently. And I just wanted to define those perspectives right now and just give you a little example question down below too. Example old diploma question. So some of these following perspectives, they'll be coming up in some of your projects and assignments too. So societal. So this perspective deals with the society as a whole. So the big group, how it affects society, people. Cultural effects. So behaviors and beliefs of a group of people. So how could you look at from a certain group's perspective? Environmental. So in this case, if you're taking the environmental perspective, you're looking at ecological and human effects on the environment. There's ethical, right versus wrong, and economic. Economic is always about money. So in this question down below, just take a quick peek at it. So we're looking at an ecological benefit and an economic benefit that are associated with genetically modified plants. Okay, so they would be numbered. Which of these? So ecological benefit. Ecological, right? Think environmental and the effects that we have on the environment. Okay. And then economic. So if we're talking about genetically modified plants, let's look at the answers here. Transfer of genetic material to pest organism. We have a decreased use of chemical insecticide. Effect on non-target organisms. Increased crop yield. So decreasing the amount of chemical insecticides would be fantastic. There's definitely an environmental impact there, ecological impact. There could be an economic one too, because it doesn't cost as much if you're not putting as much insecticide into your production costs. Effect on non-target organisms. So these are those unknown consequences. So this could be an environmental one as well. Increased crop yield. Definitely, when we're talking about crop yields, we're talking about economic money. You know, you grow more, you make more. This could also be lead to a bit of societal as well, because you're able to feed more people. So you can have quite a good conversation about all of these. The answer to this question is actually 2 and 4. So decreased use of chemical insecticides is the environmental or ecological impact, and then the increased crop yield is the economic. Alrighty, that's the end of that. There's some references on those on this presentation, and let me know if you have any problems with the practice questions.